Welcome to the Deep Dive. Today, we're unlocking the secrets of a group of organisms that are literally everywhere, yet largely unseen, and they're far more influential, and frankly, weirder than you might think. Forget just the mushrooms you pick. We're talking about fungi that craft your blue cheese, brew your beer, produce life-saving antibiotics, and even historically, inspired some pretty wild visions. Our mission for this deep dive is to give you a real shortcut into the hidden powerhouses of the fungal world, Ascomycota, often called the true fungi. We'll try to cut through some of the jargon to reveal their fundamental evolutionary tricks, how they reproduce in really surprising ways, and why their impact on everything from your dinner plate to global ecosystems is absolutely massive. Okay, let's dive straight in. When we picture fungi, we often think of a classic mushroom, right? But the Ascomycetes, our focus today, represent the absolute masters of adaptation within the fungal kingdom. They thrive in places most other life forms, even their fungal cousins, simply can't survive. So, what's the first big evolutionary move that sets these fungal superstars apart?
That's a great place to start. It really begins with their hyphae structure. So imagine a long, slender tube that's a hyphae, right, the basic building block. Now, in older fungal groups like the zygomycetes, these tubes are like open pipelines, continuous, no internal divisions. If there's damage somewhere, well, the whole system can be compromised.
Like one leak sinks the ship.
Exactly. But Ascomycota and their relatives in the Dikarya group, which also includes the basidiomycota, the typical mushroom fungi, they evolved something brilliant. Septate hyphae. Think of these as fungal pipes with built-in miniature bulkheads or cross walls. We call these septa, and they appear at regular intervals.
Okay, like compartments.
Precisely. Just like a ship with watertight compartments. If one section gets breached, the whole organism isn't immediately in trouble. Now, these septa aren't solid walls. They have tiny perforations, so cytoplasm and even nuclei can still move through. But they provide both structural rigidity and a crucial safety net. It's a huge step up in, cellular organization and protection.
That watertight compartment idea really helps. So this isn't just a structural detail. It sounds like a fundamental upgrade for survival.
Oh, absolutely. The septate structure directly leads to their incredible environmental adaptability. Dikarya can grow and fruit in much, much drier conditions than those earlier fungal groups. Some of their asexual forms are actually among the most drought-tolerant organisms known. They can thrive in environments with water activity below 0.70. That's like the osmotic pressure in jams or heavily salted fish, places most life just can't handle.
Wow, that's seriously tough.
It is. And they're also master decomposers, showing remarkable substrate exploitation. While many simpler fungi are kind of limited to sugars and starch, Ascomycetes can break down tough stuff like cellulose, and some even specialize in metabolizing keratin, the protein in our hair, skin, nails, which, as you can guess, sometimes makes them a bit of a health hazard for us.
Okay, keratin eaters, noted. Now, here's something that sounds really unique, this Dikaryon stage. Can you unpack that? It sounds complicated.
It is fascinating, yeah. So in the Dikarya life cycle, when two sexually compatible haploid nuclei, one from each parent fungus, meet inside a cell, they don't immediately fuse to form a diploid nucleus like you might expect.
Okay. What happens instead?
Instead, they pair off. They stay as two separate haploid nuclei within the same cell compartment, and they divide synchronously. So as the fungus grows, new cells are populated with these pairs of compatible haploid nuclei.
So 2 nuclei per cell, but not fused.
Exactly. This 2 nuclei per cell phase is called the Dikarya phase, and it can actually last for years in some fungi. It's not diploid, it's N plus N.
Years? Wow. Why? What's the evolutionary advantage of this elaborate dance?
It's a really clever evolutionary trick. It allows for a massive multiplication of these paired nuclei before they finally fuse, which only happens much later, just before meiosis. This means a single sexual encounter, a single initial pairing, can lead to an immense potential for genetic recombination when fusion and meiosis eventually happen.
So it's like an extended period for mixing and matching genes.
You got it. It maximizes the genetic diversity generated from that one sexual event. It gives them incredible adaptability, a way to generate lots of different genetic combinations to face changing environments or threats. It's really key to their success.
Okay, that makes sense. Very clever. So with these adaptations, how do these true fungi actually reproduce sexually? What do the structures look like? What's their signature move?
Right. The key diagnostic difference between Ascomycota and their Dikarya cousins, the basidiomycota, lies in their meiosporangia, those specialized structures that produce spores after meiosis. For Ascomycetes, these are called asci singular ascus. Think of an ascus as a tiny, often sort of cylindrical or sac-like pod. At maturity, it typically contains 8 haploid spores, and we call these
I
ascospores.
see spores. Okay.
And most asci are like tiny spore guns. They build up internal pressure to actively shoot their spores out into the air, usually through an opening at the top. There's a great example, Ascobolus, which lives on dung. It actually points its asci towards the light to make sure the spores have a clear flight path away from the dung.
Uh-huh. Aiming for liftoff. Clever. And these asci aren't just floating around, are they? They're inside bigger structures.
Exactly. The asci are usually produced within or on multicellular structures that serve as platforms for spore launch. We call these Ascomycota. And there are 4 main designs, each really representing a different strategy for getting those spores out there.
Okay.
First, you have apothecial Ascomycota. Imagine a small cup or a saucer. These have an entirely exposed fertile layer where the asci are. This allows lots of asci to discharge their spores all at once. If you, gently blow on a ripe one, you might actually see a little puff of smoke that's thousands of spores being released together. Think of those classic cup fungi.
Cool. Okay, what's next?
Second, parathecial Ascomycota. Picture a flask, sort of spherical or pear-shaped, with a narrow neck and a small opening at the top, called an ostiole. This design limits the discharge to just one or maybe a few asci at a time. You find these in many wood-inhabiting fungi.
More controlled release then.
Exactly. Third, there are pseudothecial Ascomycota. Now, visually, they might look similar to Perithecea in how they release spores, but they develop differently, and importantly, they house a specific type of ascus we'll get to, the batunicate ascus.
Okay, pseudothecia.
Yeah.
Got it. And the last one.
Last are cleistothecial Ascomycota. These are completely closed spheres, usually. No opening at all.
No opening. How do the spores get out?
Well, this often means the fungus has evolved a different dispersal strategy. The asci inside these are typically spherical and don't actively shoot their spores. They might fruit in confined spaces, like under bark or underground, think truffles. They often rely on the structure breaking down or being eaten by an animal to get the spores dispersed.
Fascinating. So the structure tells you a lot about the lifestyle. And you mentioned different types of asci, too. Those spore guns aren't all the same.
Indeed. The asci themselves show diversity in how they shoot the spores. We generally recognize 4 distinct types. First, unitunicate operculate asci. Unitunicate means single-walled, basically. These have a single functional wall with a neat little built-in lid or operculum at the tip. It just pops open and bang, the spores are ejected. You find these only in those cup-shaped apothecial Ascomycota.
Little trapdoor. Nice.
Second, unitunicate inoperculate asci. Still single-walled, but no operculum, no lid. Instead, they have a special elastic ring, like a sphincter at the tip. This ring stretches under pressure, acting like a valve to let the spores shoot through, found in parathecial Ascomycota and some apothecial ones.
Okay, pressure valve. Makes sense.
Third, prototunicate asci. These have essentially lost the ability to actively shoot spores. They're usually spherical, thin-walled, and they don't have any special opening mechanism. Their wall often just dissolves or breaks down at maturity, letting the spores kind of ooze out or just wait inside until the ascoma decays or gets ruptured. You find these in those closed cleistothecial Ascomycota or underground fungi like truffles. It's seen as a secondary adaptation, losing the shooting mechanism.
So they gave up on being cannons.
Pretty much. And finally, the batunicate, asci. Bitunicate means double-walled. This is a really cool jack-in-the-box design. They have 2 walls, a thin, rigid outer wall, and a thick, elastic inner wall. At maturity, the outer wall splits at the top, and the inner wall rapidly expands upwards, kind of like stretching out, carrying the ascospores up to the level of the opening of the ascoma to be expelled.
Whoa, like an internal piston.
Exactly. It's a clever way to get the spores out through a narrow opening, common in those pseudothecial Ascomycota we mentioned.
Okay, the diversity is incredible. But fungi aren't just about sex, right? Many have a robust asexual side, too. How does that fit into the picture?
Absolutely critical point. That's where we need the concept of the holomorph. The holomorph is the whole fungus. It includes both its sexual reproductive phase, which we call the teleomorph, that's the part that makes asci and Ascomycota, and its asexual reproductive phase, the anomorph.
Anomorph and teleomorph. Okay.
Many Ascomycetes have incredibly important asexual phases. These anomorphs often appear as what we commonly call molds. Think penicillium or aspergillus. These anomorphs reproduce rapidly and efficiently using asexual spores called mitospores because they're made via mitosis. Specifically in Ascomycetes, these are called conidia.
Conidia. And how are they made?
They're essentially modified bits of hyphae. Unlike some simpler fungi where asexual spores form inside a container or sporangium, Ascomycete conidia usually bud off as new structures or convert from existing hyphal cells.
I've heard this dual existence, the anamorph and teleomorph, has caused some naming headaches in the past. People naming the same fungus twice.
You are spot on. Historically, this was a huge issue. Because the anamorph and teleomorph often develop at different times on different substrates, maybe look completely different, mycologists often gave them separate scientific names. You'd have one name for the moldy asexual stage and another for the sexual stage producing asci without realizing they were the same organism.
That sounds confusing.
It was. Modern rules now dictate a single official name for the holomorph, usually based on the teleomorph if known. But the legacy of this dual naming still pops up and can confuse students. It really highlights how vital it is to connect these different life stages to truly understand the species. It's like trying to understand a person who uses different aliases in different parts of their life. You need the whole picture.
Makes sense. So how do scientists keep track of all these different asexual forms?
Well, we classify these anamorphs based on several features. The conidia themselves, the structures that bear them, which can be simple conidiophores or more complex structures called conidiomata, and importantly, how the conidia develop, a process called conidiogenesis. Just briefly, there are two major groups based on where the conidia form. Hyphomycetes have exposed conidiophores, like your typical fuzzy mold. Conidiomycetes form their conidia inside protective structures, often just beneath the surface of a plant host, like tiny fungal chambers releasing spores.
So it's not just what the spores look like, but how and where they're made that's key.
Precisely. Take conidiogenesis, the development process. There are two basic patterns. In blastic conidiogenesis, the young conidium is clearly recognizable. It kind of buds off before a cross wall separates it from the parent cell. Think of penicillium and aspergillus rapidly producing chains of spores this way.
Like budding yeast almost.
Sort of analogous, yeah. The other main type is phthalate conidiogenesis. Here, a cross wall forms first, partitioning off a section of hyphae, and then that section develops into a conidium. These subtle embryological details how the spore forms turn out to be much more reliable for classification than just looking at the shape of the mature spore. It reveals deeper evolutionary relationships.
Fascinating. Okay, so we've got their structure, their sex life, their asexual side. Well, quite the complex picture. But what does this all mean for us, for the world?
Oh.
Ascomycetes might be tiny, but their impact sounds huge. Let's unpack the good, the bad, and maybe the downright bizarre. Let's start with the negative side, maybe.
All right. Unfortunately, many common fungal infections in humans, what doctors call mycoses, are caused by these conidial ascomycetes. Things most people have heard of, like ringworm, jock itch, athlete's foot.
Yeah.
Those are often caused by genera like Microsporum and Trichophyton. They're specialists at attacking the keratin in our skin, hair, and nails.
And what about plants? I hear they can be devastating.
Oh, absolutely. Ascomycetes cause a huge array of plant diseases, leading to massive economic losses in agriculture and forestry. You have things like apple scab, venturia that ruins fruit, powdery mildews covering leaves, black knot on cherry trees. Then there's Dutch elm disease, Ophiostoma, transmitted by beetles, which essentially wiped out majestic elm populations across North America and Europe.
On chestnut blight.
And chestnut blight, Cophonectria, yes, which tragically eliminated the American chestnut as a major forestry. The impact can be landscape altering.
And beyond disease, food spoilage.
Yes, many common molds that spoil our stored food are Ascomycetes. But even worse, some produce potent toxins called mycotoxins. A really notorious one is aflatoxin, produced by Aspergillus flavus. It can contaminate things like peanuts and corn, and it's highly carcinogenic, known to cause liver damage. These mycotoxins are a serious ongoing threat to both human and animal health globally.
Scary stuff. It sounds like they're quite destructive.
They can be, but it's definitely not all doom and gloom. Ascomycetes are also incredibly beneficial, absolutely essential, in fact.
Okay, let's hear the good side.
Ecologically, they're premier decomposers. They're often the first colonizers of dead plant material, breaking down cellulose and other tough components, playing vital roles in carbon and nitrogen cycling in soils, forests, even streams. For instance, there's a group called aquatic hyphomycetes. They live on dead leaves and streams and make them much more nutritious for aquatic insects, forming a critical link in the food web.
So crucial recyclers.
Absolutely. And some are even predatory. Some soil hyphomyces have evolved these amazing specialized traps to catch tiny animals like nematodes, roundworms.
Whoa, predatory fungi.
Yep. And then there's food production. Many of our favorite foods owe their existence or flavor to Ascomycetes. Penicillium camemberti is responsible for the soft, ripening texture of camembert and brie cheeses. Penicillium roqueforti gives the characteristic blue veins and sharp flavor to Roquefort, Stilton, and other blue cheeses. And Aspergillus oryza is absolutely essential for making traditional soy sauce, miso, and steak.
Okay, so deliciousness. What about medicine?
Huge contributions here. The most famous, of course, is penicillin. Discovered from the mold Penicillium crucigenum, it revolutionized medicine as one of our very first powerful antibiotics, saving countless lives.
A true game changer.
And more recently, there's cyclosporine. Isolated from an ascomycete called Tolipocladium niveum, it's a selective immunosuppressant drug. It has dramatically improved the success rates of organ transplants by preventing rejection, and it also offers hope for treating autoimmune diseases. It's another massive medical breakthrough from this group.
Incredible. And biotech.
Immense potential there, too.
Yeah.
Because their genetics are relatively well understood and they can be grown easily, these conidial animorphs are workhorses in biotechnology. They can be genetically engineered to produce valuable proteins, things like human insulin, human growth factor, tissue plasminogen activator for dissolving blood clots, enzymes for cheese making, or enzymes to break down cellulose for biofuels. The possibilities are vast.
Wow. Okay, good, bad.
Yeah.
What about the bizarre?
Ah, yes, the bizarre. Well, many Ascomycetes form intimate partnerships, symbioses. Thousands form mycorrhizas, mutualistic relationships with tree roots, helping trees absorb nutrients, and then you have the lichens. These are amazing composite organisms where an Ascomycete fungus essentially domesticates algae or cyanobacteria. This partnership allows them to live in incredibly harsh environments, like on bare rock or in Arctic tundra, where neither partner could survive alone.
Lichens are fascinating. What else?
Well, maybe nothing tops the family Clavicipitaceae for sheer weirdness and sophistication and parasitism.
That sounds good.
This family includes Claviceps purpurea, the fungus that causes ergot on rye and other grasses. Eating infected grain historically caused ergotism, or St. Anthony's fire, a dreadful condition involving hallucinations, convulsions, gangrene, often death.
That's the LSD connection, right?
Exactly. Ergot fungi produce alkaloids that are precursors to LSD, and also some medically useful compounds, actually.
Okay, what else is in this family?
Then you have the genus Cordyceps. These are truly bizarre parasites, mostly of insects and spiders, but some even parasitize other fungi, like underground truffles. They invade their host, kill it, and then erupt from the body with these often large, stalk-like fungal structures called stromata. They're sometimes called vegetable caterpillars. Some are highly valued in traditional Chinese medicine.
Zombie insect fungi.
Pretty much. And their reproductive output is staggering. Some can produce something like 64 million spores from a single stroma growing out of one insect. Talk about evolutionary pressure.
Unbelievable. Okay, the diversity is just staggering. Can we maybe get a quick glimpse of the major groups just to get a handle on the sheer scale?
Sure, let's do a quick flyby of the three main subphyla, just hitting a few highlights. First, Taphrina mycotina. This includes some outliers, like Taphrina, which causes peach leaf curl. Also, here's Pneumocystis girovecii, that single-celled lung parasite causing severe pneumonia, especially in immunocompromised people, a major human health issue. And it includes the fission yeast, Schizosaccharomyces pombe, a rod-shaped yeast that divides by splitting in the middle, not budding. It's a super important model organism for genetics and cell biology, earned Nobel Prizes for researchers working with it.
Okay, subphylum 1, next.
Subphylum 2 is Saccharomycetina. This is basically the main lineage of yeasts. The order Saccharomycetales includes our familiar baking and brewing yeast, Saccharomyces cerevisiae, and also some yeasts that can be human pathogens, like Candida, mostly unicellular, experts at fermentation.
The yeast we know best. And the last one must be huge.
Yes. The third subphylum, Petsizomycotina, is by far the largest. It contains the vast majority of the Ascomycetes we've been talking about, most of the ones that form hyphae and complex Ascomycota. Within this, you have orders like the Petzizales. These are the operculate Discomycetes, the classic cup fungi with that lid-like operculum on their asci. But this order also shows amazing evolution towards enclosed underground forms like truffles, tubers, which famously use alluring scents to get mammals to dig them up and spread their spores. And it includes the highly prized edible morels, Morchella too.
Truffles and morels.
Yeah, then you have groups defined by those petunicate asci, like the Dothydiales and Pleosporales. Many important plant pathogens are here, like the apple scab fungus, Venturia. The Onygenales is interesting because it contains fungi that can digest keratin, including the dermatophytes causing ringworm and athlete's foot. Microsporum trichophyton.
The keratin eaters again.
Yep. The Uroshalus is another big one. This order includes the teleomorphs, the sexual stages of some of the most common molds on Earth. Penicillium and Aspergillus. So this connects back to antibiotics, blue cheese, food spoilage, aflatoxins, all linked here. They typically have simple, closed cleistothecia with those non-shooting prototunicate asci.
Wow, okay. It's all starting to connect. Any others?
Just a few more quick examples of the diversity. The Heliotiales are the inoperculate Ascomycetes, including things like monolinea causing brown rot on peaches, and botrytis, the gray mold, which can also be the noble rot used for making sweet dessert wines. The erysipeles are the powdery mildews, those obligate plant parasites. The hypocreales often have brightly colored paratheciae and include nectria, causes tree cankers, and apocria, whose anamorph trichoderma is widely used in biological control. We already mentioned the Clavis metaceae with ergot and cordyceps. The Sodoriales includes Neurospora, the Drosophila of the fungus world, vital for early genetics research. And the Ophiostomatails, known for causing Dutch elm disease, often have long-necked Ascomycota adapted for dispersal by bark beetles.
Woo! Okay, that's an incredible tour. We've covered a tremendous amount of ground today. From their unique septate hyphae and that really clever dikaryon strategy to the incredible diversity in their sexual structures. The Ascomycota and Answer, and then all those prolific asexual forms, the anamorphs.
Yeah.
The Ascomycota truly are a kingdom of extremes.
So what does this all mean for you, the listener? It means that this single phylum of fungi underpins so much of life on Earth. They're driving decomposition, yes, but also causing devastating diseases, forming absolutely vital symbioses like lichens and mycorrhizas, and providing us with crucial medicines and, let's face it, some really delicious foods. Their adaptability, their diverse life cycles, it's all a testament to their incredible evolutionary success.
Absolutely. So whether you're studying for a biology exam or just curious about the unseen world buzzing all around us, or maybe trying to understand how these fungal innovations impact our daily lives, we really hope this deep dive has given you a newfound appreciation for the silent yet incredibly powerful world of these true fungi. And considering the sophisticated ways Ascomycetes have evolved to adapt, reproduce, interact, sometimes with multiple forms and names, it really leaves us with an intriguing question, doesn't it? How many more fundamental secrets about life, about evolution, are still hidden in plain sight, just waiting for us to uncover them in the microbial world around us? Thank you so much for joining us on this deep dive. We hope to see you next time.